Dear students, welcome to Tungal online classes. Today we'll start with the prokaryotic cell, the structure of prokaryotic cell in detailed manner now itself. In the previous class, I have glanced over the prokaryotic as well as eukaryotic cell, what is the difference, all those things that we discussed in the previous class. So now in this class, let us start with an overview of the prokaryotic cell. In prokaryotic cell especially, there are three parts that we come across. Already I have shown you the diagram of the bacterial cell is taken as the best example for prokaryotic cell, and in that the mainly three parts that we come across. One part is cell envelope, and second one is cytoplasm, and third one is nucleoid are the three major parts that we come across over here. First one, let us start with the cell envelope. First one is cell envelope. What is this cell envelope? It is a chemically very complex envelope that is formed in prokaryotic cell. Okay, so this envelope, what it is made up of? It is made up of three layered structures. What are those three layers that we come across? One is glycocalyx, and second one is cell wall, and third one is plasma membrane, as if in a eukaryotic cell. See here, in the cell wall or in the outermost envelope of this bacterial cell, there we come across three major structures. One is the outermost glycocalyx, and inner to the glycocalyx there is a cell wall, and inner to that one the plasma membrane. This is plasma membrane. So this is the cell wall, and this one is the glycocalyx. Very important things. So then what it suggests is, it is a tightly bound three layered structure. The cell wall or cell envelope is made up of tightly bound three layered structure, right? Cell envelope is very chemically complex structure present in the bacterial cell or in prokaryotic cell, you can consider. And this cell envelope that consists of three tightly bounded layers. Three tightly bounded layers. First one is glycocalyx, and second one is cell wall, and third one is cell membrane. So there are three major parts that we come across in the envelope. What are those? Glycocalyx, cell wall, and cell membrane. Let us start with the first one, that is glycocalyx. So glycocalyx, it is a kind of polysaccharide layer. Polysaccharide layer. It's a kind of polysaccharide layer. So if it is loosely arranged, made up of poly sugar or polysaccharide, that is glycocalyx, then it is said to be as what? So then it is one type of glycocalyx that forms the outermost layer of the cellular envelope, and it is called as slime layer. Slime layer. This slimy layer, which is slimy in nature, right? So gelatinous like structure it forms. If the same glycocalyx is tough and rigid in nature, which contains capsule, right? What it is called as capsule. So slime layer is what? And capsule. And this capsule, if it is there in a bacterial cell, then that kind of bacteria is called as capsulated bacteria. What it is known as? Capsulated bacteria. If it is not there, non-capsulated bacteria. What is the difference between these two bacteria? One is capsulated bacteria always so virulent in nature. Capsule with capsule, so they are capsulated bacteria. They are known as, and non-capsulated bacteria. Capsulated bacteria are virulent in nature. These are non-virulent in nature. Virulent in the sense that cause diseases. Non-virulent in the sense that they do not cause any disease. So that is the difference between capsulated bacteria and non-capsulated bacteria. So which have capsule sometimes. Sometimes they have capsule. Then they become virulent. If they do not have capsule, then non-virulent bacteria. So this is the difference between one thing. So this is about the outermost glycocalyx. Glycocalyx. And the second one is cell wall. Let us see the cell wall now. Yes, sir, let us see cell wall. So this is lying below the glycocalyx. Where it is lying? It is lying below the glycocalyx. It is lying below the glycocalyx. And this cell wall determines the shape of the cell. It determines the shape of cell, and it is so strong and rigid one, and which gives support to the cell itself, right? And such kind of a cell wall is made of, in a bacterial cell, usually the cell wall is made of muramic acid or murane, it is called as, or peptidoglycan, it is called as. So in cells, prokaryotic cells especially, it is made up of peptidoglycan or murane or muramic acid, it is called as. So it is made up of peptidoglycan. Chemically it is made up of peptidoglycan or murane or muramic acid, it is made up of, right? So peptidoglycan, this one is made up of short strands of glycans, right? So it is made up of glycan strands. So they are very short strands of glycans. Glycans are nothing but sugars. Glycans are nothing but so glycans are nothing but sugar cells. Sugar strands it is made up of. And such kind of glycan strands make the long strand of peptidoglycan, right? Or murane or muramic acid. So such kind of strands are said to be as NAM as well as NAG. Such kind of small strands of glycans, so they form the peptidoglycan itself, right? So NAM means what? N-acetyl murane. One thing, or it is called as N-acetyl glucosamine. N-glucosamine, N-murine or melamic acid. So these are the small strands of glycans. So all together form the large strand of peptidoglycan that forms the cell wall of bacteria. So these NAM as well as NAD are bounded with the help of cross-links, and those are made up of peptide structures or peptide strands. They are these NAM and NAD are cross-linked by small peptides. Small peptides. So peptides are nothing but proteins. Small proteins. They are bounded with. So as a cross links, right? And these are playing very important role in the formation of the cell wall of the bacterial cell. So remember yourself. So this is very important thing regarding the examination. Okay, NAM and NAD. Remember. So if these are cross-linked with the help of peptides, so sometimes there are some antibiotics that we use so as to kill such kind of bacteria by lysing the cell wall of bacteria. How the lysis of the bacterial cell wall take place? It is because of antibiotics. See here, when we use the antibiotics like, so one of the antibiotic penicillin, right? Penicillin. Alexander Fleming is the name of the scientist, the one who made this penicillin, and this penicillin is the best antibiotic which lyses the wall of, which breaks the wall of the bacterial cell. Okay, how it breaks the wall of bacterial cell is it prevents or it avoids the cross-linking of these short strands of glycans, right? So with the help of peptides. So when cross-linking doesn't take place, the lysis of the cell wall that take place. So that antibiotics prevent the bacterial growth. How? By breakdown of or the lysis of the cell wall of the bacterium. Antibiotics like penicillin prevent the cross-links. Prevent the cross-links made by the peptides, right? So this is how the breakdown of the bacterial cell wall that takes place. So naturally available, one of the naturally available antibiotic like lysozyme. Lysozyme is an enzyme present in the saliva and it is also present in the tears. It is present in the saliva, it is present in the tears. So this is acting as one of the antibiotic by lysing the cell wall of the bacteria. That's why inner saliva bacteria do not exist themselves. Inner tears, so if any bacteria that falls inside the eyes, they do not exist themselves. Why? Because of the presence of this lysozyme enzyme. Very important point to be noted over here. So somewhere you may come across this enzyme. Okay. So this is about what? The cell wall of bacterial cell, right? And sometimes, so these bacteria are classified into two types. What are those two types of bacteria that we come across? On what basis they have been classified over here? So on the basis of the staining property. On the basis of a staining property, they have been classified into two types. One is gram-positive bacteria and second one is gram-negative bacteria. See here, gram-positive bacteria are those bacteria which take the stain. Stain is nothing but color. See here, gram positive bacteria and gram negative bacteria. If the bacteria which are taking the stain, what is the stain used for staining the bacteria? It is asked usually, that is crystal violet. Crystal violet is the one stain that is used for staining the bacteria. Okay, so sometimes some other stains are also used over here for staining the bacteria. So if the bacteria that takes the stain and exhibit the color, then they are said as gram-positive bacteria. If they do not take the stain and exhibit the color, then they are said as gram-negative bacteria. Gram-negative bacteria. And this property has been invented by one of the major scientists, Hans Christian Gram. Hans Christian Gram is the name of the scientist, the one who classified the bacteria on the basis of this property. Okay, so this is more important aspect to be remembered over here in case of the bacteria. One thing. One more thing is some of the Cyanophyceae members. Cyanophyta, you know it better. Cyanobacteria, you know it better. The cell wall of the cyanobacteria or Cyanophyceae is made up of cellulose, and few members have peptidoglycan as their cell wall. The point to be noted over here. Okay, cell wall of Cyanophyceae have cellulose and a few have peptidoglycan. Peptidoglycan. Point to be noted over here as far as it is concerned. Okay, so coming to the next one, cell membrane. This is plasma membrane, which is similar to plasma membrane in eukaryotes. Similar to the plasma membrane in eukaryotes. Okay, so what is this cell membrane? It is a thin membrane which lies below the cell wall. See here, this is cell wall, and below the cell wall what is there? So cell membrane is there. It lies below the cell wall. It lies below the cell wall. Very important point. So it is semi-permeable. It is semi-permeable in nature. It is semi-permeable membrane. It allows only restricted materials to enter in and exit out. So that is why it is said to be semi-permeable in nature. Semi-permeable in nature. One more thing. Come to the next point. Sometimes, so this cell membrane itself makes some of the invaginations, and such invaginations made by the cell membrane are called as mesosomes. What they are known as mesosomes. So already I might have draw this one, but so it doesn't create any interest. See here, when I bend like this, now you will come to know this structure very clearly, that is mesosomes. So this is one of the unique features of bacterial cell or the prokaryotic cell. It makes some invaginations. And those invaginations are called as mesosomes. mesosomes. What they are called as mesosomes, right? And these mesosomes, so they are of different types. These mesosomes, among them, one is a septal mesosome. One is septal mesosome, and sometimes some more type of mesosomes that we come across over here that forms different kinds of shapes over here, and one of the mesosomes which is very nearer to the nucleoid, right? So one more thing you must remember, one of the mesosome is nearer to the nucleoid. That helps in duplication of DNA during bacterial cell division, right? So mesosomes, septal mesosomes, they help in making the cell wall, right? So cell wall, they help in making, right? So this is very important point. And enterosacular, so mesosome is also there. There the sac-like structure is formed over here, and that kind of mesosome that has some of the respiratory pigments within it. Some of the respiratory pigments are present within these mesosomes, and hence it helps in respiration. Hence it helps in respiration, so that it is equivalent to one cell organelle in case of eukaryotic cell. This is prokaryotic cell actually. It doesn't have any membrane bound cell organelles like mitochondria already I have mentioned. But the cellular respiration must take place over here. That respiration is done by these mesosomes. Hence this act as, mesosome act as mitochondria. This act as the mitochondria. Very important point to be noted over here as far as meat is confined. So see here, so mitochondria, they are acting as, means they help for cellular respiration, energy production itself, right? So this is more important aspect to be remembered over here. Some of the mesosomes which have respiratory pigments, they act as the mitochondria, right? So mesosomes. Some mesosomes are vesicular in nature, lamellar or lamellae type, right? So some are tubular, right? So what is the major function of mesosome if you are asked? Then they help in making the cell walls, they help in the respiration, and sometimes they help in increasing the surface area of plasma membrane. Very important point. So if such kind of bendings are seen here and there inside this one, then it increases the surface area, it helps in secretion, it also helps in secretion of some substances. It helps in secretion also. Okay, so that's why these are the major functions of this what? Mesosomes. Mesosomes help in cell wall formation, respiration, secretion, right? So this is the use of these mesosomes. Mesosome is one of the important feature of this bacterial cell or the prokaryotic cell. This is about the cell membrane. So all the three layers that we discussed, one is peptidoglycocalyx is there, cell wall is there, cell membrane is there. All the three are forming the cell envelope itself. They are tightly arranged. Even though they are tightly arranged, they perform different kinds of functions. Point to be noted. So a very important point, it is related with the cell envelope. Okay, this is all about the cell envelope. And what is the next structure? Already I have written over here. What is the next structure? Next structure is the cytoplasm. What is this cytoplasm? So what it contains, all those things let us study now itself. Before the study of the cytoplasm in bacterial cell or in prokaryotic cell, let us see flagella itself, locomotory organs in these bacterial cells and other structures that we come across over here. What are those structures? Let us see one by one. First of all, we will start with the flagella or flagella, right? So these flagella are in pairs or in clusters, right? In bacteria, different types of bacteria are there based on the arrangement of these flagella, right? So you know it better, you have classified already in the previous class, I hope. And the next one is flagella. It is a locomotory organ in bacterial cell or in prokaryotic cell. And this flagella, it is a whip-like, right? It is a whip-like structure, so that helps in locomotion of bacteria. Okay, and these are made up of a protein called as flagellin. These are made up of a protein called as flagellin in prokaryotes. So, and this flagella has, so there are three parts that we come across in flagella. What are those three parts that we come across in flagella? So flagella has first one, that is filament. Filament is the first one. And second one is hook, and third one is basal body. These are three major parts that we come across, and whenever we see the size of these these are 0.01 micrometer to 0.02 micrometer in diameter, and they are 0.3 micrometer to 20 micrometer in length. So this is of whip-like structures that help in locomotion of these bacterial cells, right? So it has three parts. One is filament, it is the longest part of this flagella. Hook, so it is a bent tubular structure that connects, a bent tubular structure that connects the basal body to filament. And basal body is nothing but it is the proteinaceous structure, blepharoplast it is called as, and that blepharoplast is the basal body. How its structure is? Let us see the structure of this flagellum. So there is a long rod-like structure that attached to cytoplasmic membrane, that attached to the cytoplasmic membrane. And here where you come across at the base, there is one ring-like structure that you come across. This is said as M ring. Above this one more ring that you come across. This is S ring. And above this one, two more rings that you come across here. Right? So this is here P ring is one more ring and L ring is one more ring itself. That we come across two pairs of rings. All these together form the basal body. Right? And this one is the ring, means here you come across the structure. This ring is made up of lipopolysaccharide layer. Lipopolysaccharide layer it is made up, and this layer is made up of peptidoglycan layer. The entire structure together form the basal body, and this bent tubular structure is called as hook. It is hook that links the basal body to the filament. This structure itself is called as filament. You remember all these rings in the basal body that you come across, and hook as well as filament. So all these three together form what? Entire the flagellum itself, right? So this is the structure of the basal body, hook as well as filament. All these three together form the structure called as flagellum. One flagellum, right? So this is one locomotory organ that we come across. Along with this one, some other structures that we come across here, what they are said to be as, so this kind of, so extensions that you come across, they are called as pili. What they are known as pili. Okay, so pili as well as fimbriae are the structures that we come across over here. Yes, now pili as well as fimbriae. Pili and fimbriae that we studied over here. So these are also the appendages. Appendages present along the flagella. These are also, so the appendages present along the flagella in the cellular envelope. In cell envelope. In cell envelope they are present in. And first of all we will start with the pili. So these are hollow, cylindrical, filamentous, short structures than that of flagellates, right? And they do not help in locomotion. These do not help in locomotion. So hence these are present in both non-motile and motile bacteria. These are present both in the non-motile as well as motile bacteria. They do not help in a locomotion, of course. They are not the locomotory organs. They are hollow cylindrical extensions that arise from the cellular envelopes, and sometimes some of the pili help in conjugation between two bacterial cells. See here, this is one bacterial cell, this is another bacterial cell. So these two are linked with the help of a linkage called as conjugation tube, right? During reproduction in two bacteria, one is positive strained bacteria and another one is negative strained bacteria that I consider. So in bacterial reproduction, there are three types of reproduction that occurs. One of them is conjugation method, right? So exchange of the genetical material that take place between two strained bacteria. One is positive strain, another one is negative strained bacteria. So the transfer of the genetical material that take place through this conjugation tube itself, and that conjugation tube is made by the pili is called as sex pili. Remember, sex pili. They help in making the conjugation tube between two strained bacteria during reproduction. During reproduction, they are called as sex pili. So sex pili are very limited in number, right? So this is the fact what you should know over here itself, right? So this is sex pili, it is called as. So this is about the pili, right? What about the fimbriae? So these fimbriae are Bristol-like appendages. These are Bristol-like appendages present along with these pili. So they are shorter than the flagella and they do not help in locomotion. So this is about some explanation regarding the fimbriae. These fimbriae are also made up of, means they are made up of proteins. That is helically arranged proteins they are made up of, and pili are also made up of the protein called as pillin. Right? One more point we noted here. So pillin is the protein these pili are made up of, and fimbriae are also made up of the proteins which are helically arranged proteins. Right? This is about the pili as well as fimbriae along with the flagella. All these are the components of the cell envelope itself, right? So they are coming out of the cell from the cell envelope itself. That's why I have added these structures in the cellular envelope structures itself. After this one, let us go to the next point. So very important point, that is cytoplasm, and after that one, nucleoid, right? Yes, cytoplasm. Next one is cytoplasm. It is a semi-fluid matrix present inside or lying inside the plasma membrane. Lying inside the plasma membrane that contains many number of cell organelles. It contains cell organelles like ribosomes, right? So, and one more important structure that we come across over here. So, what is that? Inclusion bodies, right? So, let us go to the ribosomes first and later inclusion bodies. So, what are these ribosomes? So ribosomes are nothing but they are membraneless. These are membraneless cell organelles. These are membraneless cell organelles, and these are made up of two subunits. How many subunits they are made up of? Two subunits. And these two subunits, wherever you see diagrammatically this kind of structure, and the type of ribosomes present in prokaryotic cell is 70S type of ribosome. What type of ribosome it is? 70S type of ribosome. It is 50S subunit and it is 30S subunit. Like this, there are two subunits that we come across over here. Total it is 70s subunit. So 70s ribosome it is called as. So this is more important point to be noted over here, right? So in prokaryotic cell, 70s type of ribosomes are present. It has two subunits, one is 50s and 30s. And there are two types of ribosomes that we come across. One is bounded ribosomes, and the second one is free ribosomes. Bounded ribosomes are associated with the plasma membrane. See here, they are bounded to the plasma membrane. They are attached to the plasma membrane. Sometimes they are freely selling in the cytoplasm. These are also ribosomes called as free ribosomes. Freely selling the watery substance, that is semi-fluid matrix itself, is called as cytoplasm. Okay. And free ribosomes, they are called as, and one more unique feature is there in prokaryotic cell. What is that? So that is polyribosomes. Very important feature. What is this polyribosomes? See here, a chain of ribosomes are seen over here in the diagram. If all these ribosomes are bounded with the help of, or they are bounded to only one mRNA, what they are bounded to, what is this structure? mRNA. See here, this is one ribosome, this is another ribosome, this is another ribosome. All these are bounded to the single mRNA. This structure itself is mRNA, messenger RNA. Messenger RNA. So these are ribosomal subunits, right? These ribosomes are many in number. They are bounded to only one ribosome, messenger RNA itself, right? So, but one thing you should remember: one ribosome to which one mRNA is always bounded, so as to synthesize one protein, right? Point to be noted. One mRNA helps in synthesis of one protein, right, which is bonded to the ribosome. Okay, here many ribosomes are there. They are synthesizing what? Protein, of course. The same copy of the protein they are going to synthesize. Means they are going to synthesize many copies of the same protein. See here, these polyribosomes, they synthesize many copies of same protein, right? So this is the fact what you should remember. This is very important for read again. Very important one point to be noted. This is all about the what? The ribosomes. One. And the next one is inclusion bodies. What are these inclusion bodies? They are the small bodies without any membrane. So they have, and they are membraneless inclusion bodies. So they are present both as organic as well as inorganic form. Let us see them now. Yes, inclusion bodies. These are reserve material storing bodies. So these are reserve material storing bodies. These are also said to be as storage reserve granules, and these are not bounded with any membrane, and these are they lie free in cytoplasm. They lie freely in cytoplasm, and these are sometimes organic granules or inorganic granules. These are in two forms. One is organic granules and inorganic granules, right? So there are two types of granules that we come across. Organic one are phosphate granules and synophysian granules and glycogen granules are the best example for this one. Organic, phosphate and synophysian and glycogen granules are the best example for this organic one. Inorganic one, first one is volutin granules. Volutin granules are one kind of granules which are said to be as inorganic granules, and these take stains. So these are the granules which take stains. Some of themselves are sulfate granules, even sulfur granules. They are sulfur granules, and these are said to be as metachromatic granules. Why? Because they take stain, right? So these are the different types of inclusion bodies that are seen in case of the bacterial cell or the prokaryotic cell. In the cytoplasm, these are freely lying, and the next one is very important one, that is nucleoid over here. Let us see the nucleoid in detail now. Yes, third important structure is nucleoid. Okay, in bacterial cell, there you come across the chromatin network or the chromosomes or the genomic DNA. It is called as, it is not at all surrounded with any nuclear membrane. Hence, we can say that there is no definite nucleus, right? It is naked DNA. We can call it as. Okay, so in prokaryotes, there is no definite DNA, there is no definite nucleus, since the nucleus is not surrounded by nuclear membrane. Hence it is called as naked DNA. What it is called as? Naked DNA or pronucleus, first formed nucleus, or it is called as incipient nucleus, right? So incipient nucleus, like this. Many names are there for this nucleoid. It is also known as nucleoid. It is also known as nucleoid. Nucleus containing material is there, but the nucleoid, so nucleus is not there. Why? Because it doesn't have any definite boundary or nuclear membrane around the nucleus. So this is very important aspect to be remembered. So this is also consisting of the chromosomes, means it also makes chromosomes, and it also contains genes, and it is called a genomic DNA. It is also known as, right? So it is circular in nature sometimes. What happens from here? So here you are observing some of the circular structures. These are nothing but plasmids. These plasmids are nothing but they are the circular DNAs, right? So these are lying away from this DNA. they are not confined to these chromosomes which are formed as a result of condensation of the chromatin network. So hence they are extra chromosomal. Self-replicating means they can duplicate themselves on their own. Self-replicating circular DNAs. What they are named as? So they are extra chromosomal, self-replicating chromosomes or self-replicating circular DNAs. So plasmids. How to define these plasmids? These are extra chromosomal self-replicating circular DNAs. Circular DNAs, right? So these are lying away from the genomic DNA, but still these are playing very important role and they are very much helpful for the human being nowadays in biotechnology. So as you study biotechnology in detail, that moment you will come to know. So but remember here, these are the plasmids which are of different types in different bacterial strains, and those plasmids are acting as the vectors. What is plasmids act as? They act as vectors. Vectors in the sense that, so vectors are those structures which can carry the DNA, other DNA or foreign DNA from one organism to another organism. So they help in carrying foreign DNA, right? They help in carrying the foreign DNA from one organism to another organism. Such kind of structures are known as vectors. Means they are carriers of genes, carriers of the DNA, right? They act as, and these are used in rDNA technology. These are used in rDNA technology, recombinant DNA technology, for bringing about some of the good products for the welfare of the human being. Example is insulin, right? So as a result of rDNA technology, insulin is produced over here nowadays by using genetic engineering. What it is said as genetic engineering. It is utilized, right? So that is the use of these plasmids, right? So plasmids are nothing but extra chromosomal self-replicating circular DNAs, right? These are also present over here inside the bacterial cell or the prokaryotic cell. This is all about the prokaryotic cell. Explanation about the prokaryotic cell, right? So these are all things that I have covered in prokaryotic cell, and next one is eukaryotic cell. Let us discuss it in the next class. Thank you.